Do subscribe to Ekeeda channel and press bell icon to get updates about latest engineering, HSC and IIT JEE main and advanced videos.
Hello students, today we are studying chapter Gene, its nature, expression and regulation. In that we are studying topic DNA is genetic material. Now various experiments like Griffith, Lenderson's proved that DNA could be genetic material, but one experiment of Hershey and Chase ultimately proved that DNA was the genetic material. Let's study about it today. DNA was the genetic material present in human beings or prokaryotes, eukaryotes. In certain prokaryotes and in all eukaryotes. Now this was passed down from your parents to you, and this was proved by Hershey and Chase. For that experiment they used T2 bacteriophage. Now T2 bacteriophage is a virus that infects bacteria. Now one parameter that was striking thing in that was DNA basically has phosphorus and it never contains sulfur, whereas your proteins have opposite of that. They have sulfur but they never had phosphorus. So how the experiment was carried out? It was carried out in two sets. In first set, the bacteriophage protein coat was radioactively labeled and the phage infected the bacteria. Once it does, there was no radioactivity involved. So proteins were ultimately not passed down to the phage. But in other case, when the phage DNA was labeled and the phage was infected to the bacteria, what they observed was that radioactivity was observed from the bacteria which was centrifuge. Ultimately, it laid down the theory that DNA was the genetic material that was actually passed. DNA is the genetic material. Now DNA is the genetic material that is present in everyone's cell. It is passed on to everyone from their parents, mother and father. Now Hershey and Chase went to prove this using T2 bacteriophage. Now bacteriophages are the viruses that infects bacteria and a bacterial strain. The key point was that the DNA has phosphorus but it never contains sulfur. Whereas your proteins have sulfur in them, their polypeptide bonds basically have, but they never contain phosphorus. So in the first part of the experiment, the protein code of the bacteriophage were radioactively labeled. In the second part, the phage infected the bacteria, but after radioactivity was never seen in the bacterial cells once it was centrifuged. Whereas in the part 2 of the experiment, the phage was DNA labeled. The DNA of the phage was radio-labeled and it was made to infect the bacterial strain. But once the phage infected the bacterial strain, what was observed was radioactivity. It means the DNA was transferred to the bacterial strain. It means DNA was transferred and the centrifuge phage showed radioactivity. Students, in this part of the chapter, we did study about how DNA was the genetic material. Thank you.